Welcome to Learn Microbiology to another video on the series Cell Cycle. In this video, we will be studying about the regulation of the cell cycle. Let's get started. First, let us understand why there is a need for regulation control. This is a cell cycle which we studied in the previous video. In case you have not watched the previous video, I will leave the link in the description below. Let us consider one cell over here. Now let us consider that this cell is committed for the cell cycle and it is going to start the cell cycle which consists of your G1, S, G2 and M phase. After first cycle, we can see the cell divides into two. After second cycle into four, and this continues. Okay, so can you just count the number of cells? We just have a very simple formula to calculate the number of cells, that is 2 raised to n. The number of cell cycle was 7, so 2 raised to 7 will give us 128 cells. So as you observe here, just in a matter of 7 cycles, we get 128 cells. What if I didn't stop the cell cycle? 128 cells will get doubled in the next cell cycle. And if this goes on, this creates something known as human or cancer. Secondly, the cell cycle is unidirectional. If there is no control, it is possible that cell cycle will return back and after G1 goes to M phase, and that is going to create a devastating effect. Thirdly, we know that DNA replication takes place in the S phase. For example, if there was no control, then it is possible that before the completion of DNA replication, the cell can go to the G2 phase and then to the M phase. What will happen because of that? The cell which divides will not receive a complete set of the DNA. So to avoid all these issues, we have something which is called as this regulation control. We will be studying about regulation control in detail in this video. Cell cycle is controlled by certain protein complexes which are known as cyclins and CDK. Let us understand about them in detail. This green color diagram represents our cyclin and this particular pie represents our CDK. Now why it is named as cyclin? Because this protein molecule is going to undergo a series of synthesis and degradation. CDK is known as cyclin dependent kinases. Kinase enzyme adds a phosphate group to a target protein, and this kinase enzyme is dependent on cyclin, hence the name CDK. Cyclin has a regulatory role while your CDK has a catalytic role. We have different types of cyclins which are present at different phases of the cell cycle. We have G1 cyclin which is active during your G1 phase, the G1S cyclin which is active during the late G1 and the early S phase, the S cyclin which is active during the S phase, and the M cyclin which is active during the M phase. Now let us understand the Cyclin-CDK complexes which are present in vertebrates. We learned that there are G1 cyclins, G1S cyclins, S cyclins and M cyclins. All these are common names. In different organisms, you can have a defined name. So let us learn what they are known in vertebrates. The G1 cyclin is known as cyclin D, which is bound to CDK 4 and 6. G1S cyclin is known as cyclin E, which is bound to the CDK 2. The S cyclin is known as cyclin A, which is bound to CDK 1 or CDK 2. Then the cyclin M is known as cyclin B, which is bound to CDK 1. All these cyclins are present in vertebrates throughout the different phases of the cell cycle. Having understood the types of cyclins, now let us move towards understanding how these cyclin and CDK molecules are regulated. Cyclin CDK regulation. The first step of regulation is by the synthesis and degradation cycle of cyclins. This is going to be at the level of cyclin. Let us have a look at this particular graph. At the y-axis, we have the concentration of the particular cyclin. X-axis, we are having our G1 phase, S phase, G2 phase and your M phase, that is mitosis. As we can see here, initially your cyclin D synthesis increases and then it becomes constant throughout your S phase. Once S phase is completed, you can see your cyclin D is degraded. Then by the end of G1 phase, the synthesis of cyclin E starts, which is at a peak during the transition of G1 to S phase, and it is degraded further in your S phase. By the end of G1 phase, the synthesis of cyclin A also starts, and at G2 phase it is having a peak value and it is going to be degraded once the cell moves towards the M phase. The cyclin B, which is your M cyclin, which synthesis starts during the S phase, it goes on increasing and it becomes maximum when the cell moves from the G2 to the M phase, and in the M phase it is degraded. Thus we can see all the cyclins are synthesized and degraded at different rates during the progression of the cell cycle. They play a very important role for the cell progression. For example, as we can see, once the cell moves from G1 to S phase, your cyclin E is getting degraded. This will give a signal to your cyclin A, whose synthesis then begins to increase. And once the cyclin A starts degrading, this gives a signal to your cyclin B and its synthesis increases. And this particular coordinated mechanism will help in your cell progression from one phase to the another phase. Now, synthesis takes place by transcription and translation of the cyclin genes. We already know there are certain regulations even at that level, and the most important regulation is at the degradation level. Cyclin B is degraded with the help of ubiquitin dependent proteolysis mechanism, which is catalyzed by the enzyme E3 ubiquitin ligase. Ubiquitin ligase enzyme, there are two types. One is your SFC, which is a ligase enzyme which contains three protein components, the SKP1, the Cylin and F box, hence the name SCF. Now this SCF ligase enzyme is going to break down your G1 cyclins. So in the case of vertebrates, they are your cyclin D. Secondly, another E3 ubiquitin ligase enzyme is your APCC, which is known as anaphase promoting complex or cyclosome. This is going to degrade your S cyclins, that is your cyclin A, and M cyclins, that is your cyclin B. Thus, when these ligase enzymes are present, they will degrade your cyclins which are present at the different phases of the cell cycle. I hope the first level of Cyclin CDK regulation is understood. Let's move to the second level. The second level of Cyclin CDK regulation is concerned with your CDK molecule. The mechanism is known as phosphorylation dephosphorylation. Let us take the example of M-CDK, that is a CDK molecule which is bound to your M cyclin. Once cyclin and CDK binds together, the molecule doesn't become active. There is one enzyme called as V1, which is a kinase enzyme which is going to add phosphate to your CDK molecule at the tyrosine at 15th position or threonine at the 14th position. So once this phosphate is added at this 14th or 15th position, your CDK molecule will become inactive because this phosphate is an inhibitory phosphate. So we need to activate this complex. We require another enzyme called as the CAC or the CDK activating kinase. This particular enzyme is going to add a phosphate at the threonine at position 161. And this phosphate is known as the activating phosphate. So we expect that once activating phosphate is added, our molecule should have become active. But that doesn't happen. Our complex is still inactive. Why? Because the inhibitory phosphate is still prevalent. If we have to activate our complex, then we have to remove this inhibitory phosphate. and this done by the CDC25, which is going to remove the phosphate. It is a phosphatase enzyme, and now our complex is active. Now to make this easy for you to understand, let me give you an analogy. The V1 enzyme is going to inactivate your complex, right? By default, the complex is inactive. Let me give you an example. Consider you going to your college. Before entering your college, you have to pass through the college gate, which is locked, that is, which is inactive. Why? Because it is locked. Then when you show your identity card to your security guard at the gate, then he will allow you inside. But you can only go inside once the gate lock is opened. So that's why you can now have your ID card, that is your activating phosphate. Now you can take an active participation in your college activity. I hope this particular analogy will help you in remembering this mechanism. Let us move to the third type of regulation. The third level of regulation is also concerned with your CDK, that is binding of CDK inhibitor proteins. This is our CDK cyclin complex, which is in the active form because it has got the activating protein. There is one molecule called as CKI, which is a CDK inhibitor protein. Now when this molecule binds to your active form of your complex will become inactive. Let us understand in detail about this CKI. CKIs have two families of proteins. One is your CIP-KIP family and another one is your INK4 family. CIP-KIP means CDK interacting protein or kinase inhibitory protein, while INK4 means inhibitor of kinase 4. The proteins belonging to these families are P21, P27, P57 belongs to the CIP-KIP family, while P16, P15, P18, P19 belongs to your INK4 families. These protein molecules are nothing but your CDK inhibitor proteins, which will bind to a particular CDK molecule and inactivate it. Your P21, P27 and P57 molecule binds to cyclin CDK2, while P16, P15, P18 and P19 protein molecule will bind to cyclin CDK4/6. So, once this particular inhibitory protein binds to the particular CDK, our molecule becomes inactive. So, that is our third level of regulation. Let's revise what all things we studied in this video. First, we understood that the cell cycle needs to be regulated. Secondly, it is regulated with the help of complexes called as your cyclin CDK molecules. Then we understood that there are three levels of regulation of your cyclin CDK complexes. In the first level, where synthesis and degradation of cyclin takes place, degradation takes place with the help of ubiquitin ligase enzyme like your SEF and APCC, and this molecule is going to degrade a particular cyclin. Secondly, we understood the second mechanism was phosphorylation dephosphorylation of the CDK molecule. This occurs with the help of V1, CAC and CDC25. Unless and until the CDC25 phosphatase removes your inhibitory phosphate, your complex will not get activated. Thirdly, we understood the binding of CDK inhibitor, this particular protein molecule which belongs to the two types of families, binds to the particular CDK cyclin complex, it will inactivate it. And students, in today's video we studied a very important topic which is going to help you in solving many questions of your entrance examination. If this video was helpful, do share it to your friends, like the video, do subscribe to it so that you will be notified whenever I upload a new video. Thanks for watching this video. I'll see you all in the next video.